So this is a relatively new agent. Do you mind taking me through this study and what it tells us about it and how it could be effective for diabetic cardiomyopathy?
Yes, absolutely. So the molecule that we're studying is an inhibitor of an enzyme which is called aldose reductase. Aldose reductase is a very critical enzyme from a metabolic point of view, critical in the least favorable interpretation of the word critical, right? Critical is a word that could mean many different things in the sense that it doesn't do much good. It transforms glucose into a toxic. And the reason probably it does that is in the attempt of removing glucose. In patients with diabetes, you have an excess of glucose, and that per se activates an enzyme that transforms glucose into something else. Unfortunately, that something else is as bad, if not worse, than the product you started from. It's called sorbitol. Sorbitol cannot cross the membrane of cells, so gets stuck inside. And because of that, cells could be progressively damaged or acutely damaged and die. In patients with diabetes, this mechanism of abnormal utilization of glucose takes place in different cells in the body, including the cardiomyocyte, which are the cells of your heart. And if they do so, they get damaged, you can lose them, and they get replaced. They get replaced not by sister cells, but rather by less capable cells, like a scarring tissue, like when you get a cut. Your skin doesn't really actually restore an identical patch. You have a patching, right? Unfortunately, the patching doesn't have the same property of the original. And the problem from a heart function point of view is that that ability of contracting is compromised. And that's really the premises for overt heart failure, which doesn't mean anything other than the failure of your heart to function as a muscle. So the study that we conducted aimed at verifying that the use of the drug was safe and well tolerated. And the result of the study clearly showed that. And in addition to that, the study explored whether there were changes in biomarkers that could be indicative of a potential benefit from a cardiological point of view, suggesting that there is signal in the right direction. And this was the basis for designing the formal phase 3 outcome study that will study patients with type 2 diabetes. They already have changes in their heart. They're changes that you could document by an echocardiography from a structural point of view, also from a functional point of view. And patients with those characteristics will be followed over time, 15 months, to see if treatment with this molecule, which is called AT-001, changes the slope of decline. And that is measured by the functional capacity. Functional capacity means exactly what it sounds like. What is your capacity to function? So there is a machine that measures how much oxygen you need to do certain exercise, and the amount of oxygen you use is an indicator how much exercise you're capable of performing. So patients are screened at study start to see what is their functional capacity and at study end. And the premise is that the placebo group will continue to decline, where patients treated may do better. The design contemplates 2 doses for AT-001 versus one group that is on placebo.
Now, just based on the results of this study, what are the next plans to examine AT-001 in a larger patient group and to more fully evaluate the drug?
Yeah, so the study I alluded to, the phase 3 study, which is called ARISE-HF, will investigate whether we could provide long-term benefit by changing their deterioration of functional capacity. This patient, if not managed, will continue to decline. So the objective of this study will be to investigate whether you could alter that. The study will also have an extension to see whether as a result of this modification of the natural course of the disease, the fatality of the development of so-called overt heart failure are reduced in the patient exposed to AT-001. So that is the biggest program that we have initiated. In parallel, we will be conducting what people call mechanistic study that will investigate the mechanism by which AT-001 may provide benefit. What does AT-001 do to the utilization of fuel for energy production by the heart cell? Does help them to utilize correctly glucose, or continues to force these cells to utilizing other substrates? So that's more of a mechanistic study, is trying to understand the reason why it may provide benefit. I think is an important element of scientific knowledge that will contribute to understand why the drug may provide benefit.
Yeah, that's really interesting. I think the mechanism behind it is something people will be very curious about, especially because in this safety trial, we didn't have any adverse events that led to discontinuation based off the data that I'm seeing. And there was also no, I don't want to get this wrong, there were no reported changes or no clinically relevant changes reported in liver and kidney function. How much of a good sign is that for an agent like this, that it appears to be as safe as it is?
Yeah, so in general, it's an excellent sign, right? So when dealing with a large population and with what people call a common disease, a disease that is widely spread, you really want to make sure that the safety record is excellent, right? Just because there will be so many people exposed. And in this case was excellent. Now, so in addition to these theoretical values, always so nice to have an impeccable record. When a few decades ago, this molecular target was identified for drug development, the challenge that the previous company developing molecule for this target faced, they were not able to identify molecules that would be sufficiently safe while efficacious. So in our case, we started off by understanding why they failed. So we started off by trying to see why the previous compounds did not succeed by redoing the crystallography of the enzyme, characterizing the enzyme, and identifying molecules that would be selective for this enzyme without cross-reactivity with other molecular pathway. And some of the other study we will be showing at the ADA specifically look at that, not just theoretically, not just as an assumption, but really doing head-to-head comparison with those molecules that were toxic. And they clearly showed that the compound called AT-001, while hitting the target enzyme, which is called aldose reductase, they do not hit other potential pathways that are very relevant and that, if interacted by, will have a toxic effect. So rather than just base this on an assumption, we decided to carry on the study directly. And it's a head-to-head study. and clearly shows the specificity and selectivity of AT-001 when compared to some of the old molecules.
Questions. Was there anything else you wanted to add that you think is relevant for clinicians to take into consideration?
And maybe the only thing I like to add is that diabetic cardiomyopathy is very relevant for patients with diabetes. Patients with diabetes have more than one problem they may be facing in their life. But as far as their cardiological well-being is concerned, they're exposed to atherosclerosis, so changes in the coronary artery permeability. And that is what we have learned for years, right? So we do know that the coronary artery could get changes similar to patients with other medical conditions, right? Or just as people get old. But what we're looking at is another potential complications of patients with diabetes, which is the ability of the heart to function as a muscle. So the heart has to compress and push blood that goes from the atrium to the ventricles and then to the aorta. So that is the ability of the functional capability of your heart as a muscle. And we keep losing patients with diabetes for heart failure. So heart failure is a major concern for patients with diabetes. And obviously, what we have today is not sufficient. For diabetic cardiomyopathy, absolutely nothing. There are no therapies today. We only have therapies for patients that have so advanced and have progressed so far, for which their heart is no longer compensating, can no longer handle the volume of blood they need to manage. So what we're trying to do, we try to reduce the volume of blood the heart muscle has to handle. So this therapeutic approach target a very unneeded, very unmet medical need that accounts for about 20-25% of the patient population with diabetes. So it's a very relevant condition.